Hello friends, welcome back to my channel Biofric. In today's lecture video, I'll be talking about prokaryotic replication, that is in E. coli. So in E. coli, replication starts at origin of replication known as ORI-C, which is about 245 base pair long sequence. Now this particular ORI-C, origin of replication, has 9 more repeats, that is 9 base pair sequence that is repeating there for about 4 to 5 times. The repeat 9 more that you can see in brown color, and green ones is 13 more repeats, that's 80 reach. And this 13 more repeat is nothing else but this is the 13 base pair sequence repeating for about 3 times. Now the first thing that occurs in the replication process, the initiation of replication begins with DNA A that takes ATP along with it and binds to 9 more repeat. Along with that, there comes the factors like HU and IHF that will bind to the DNA, and DNA will slightly bend now. As the DNA bends, helicases like DNA B, okay, in prokaryotes, the helicase is DNA B and it is loaded by DNA C. It requires this DNA C so that it is loaded onto the DNA, and helicase does the job of melting the DNA. So DNA is melted near the 13-mer region, because 13-mer region is 80 rich and it's easy to melt that particular region rather than GC rich region, right? So once the helicase starts unwinding the DNA, single strand binding proteins bind to this single strand that's formed, okay, so that it doesn't bind to its complementary single strand and form the duplex again, because we need that single strand so that it synthesizes, the polymerase synthesizes the complementary strand and you get the replicated DNA there, right? And as the DNA helicase melts the DNA, upstream and the downstream of that replication fork, there'll be the strain that is caused, okay, there'll be supercoiling them. So that has to be relieved by DNA gyrase here, that is topoisomerase 2, and that's how it proceeds. Now DNA G is the one that is important for the lagging strand because it will add the primers. It has primase activity, okay. So it has this transient interaction with the helicase, that is DNA B, and it will start adding primers onto the lagging strand. Now how this elongation takes place? Okay, so we know that DNA G is adding primer there. It has to interact with the core pol 3, that is polymerase 3 enzyme there, okay, and it will, this particular interaction will limit the primer length. It shouldn't be the case that whole lagging strand has complementary primer nucleotides, okay, it shouldn't be that particular thing, right? It has to stop somewhere and later polymerase has to take over the job of polymerizing the DNA strand complementary to that single strand. So it also, this particular interaction facilitates the exposure of this 3' end, okay, because 3' end, 3' OH group of the primer is important, because that's the sole idea of adding primers. Polymerase requires that 3' OH so that it can polymerize the further, synthesize the DNA later, right? So on lagging strand, what happens is the beta clamp. Okay, before that, let's look at the polymerase 3 enzyme, okay. In the right bottom side, I have drawn the polymerase 3 enzyme, wherein there is alpha, epsilon and theta subunits, which are the co-proteins, okay. So alpha subunit has polymerase activity, epsilon has proofreading activity, theta helps epsilon subunit for proofreading activity, okay. Now there is also the tau subunit that is holding this core protein together. Now the left side beta clamp is important for the polymerize. It will hold the leading strand, while the right side beta clamp, where I have written the lagging, on top of which we can see the gamma and delta subunits, okay. These are holding the lagging strand, okay. So now the beta clamp, okay, for lagging strand, okay, is requires the clamp loader. Now this clamp loader is gamma subunit. So beta clamp is loaded by gamma clamp loader complex, okay, onto the primer template junction. So wherever it finds that primer template junction, that will be loaded there. And now what happens is polymerase 3 can cover that portion. It will synthesize the complementary DNA. Now again, the clamp loader will hold catch hold of another primer there. It will give to the clamp, and then the polymerase will polymerize the next batch. Okay, so that's how you will get Okazaki fragments. Okay, so Pol 3 transits from previously completed Okazaki fragment to the new primer. So you can imagine that there are primers in between. Clamp holder there. Okay, the sliding clamp is catching hold of the DNA from that primer there, and then polymerase will carry out its job. Okay, so as the polymerase activity is going on, DNA G primase will leave. Okay, and that's the continuous process. Like that, you will find Okazaki fragments. Now polymerase 3, it will keep on synthesizing the new fragment. Okay, and it will stop near the uh, end of the previous Okazaki fragment, because it has this starting primer there, right? So it will stop there. Now how does the termination procedure occurs? Okay, so terminator binding protein, that is TUS protein, is important here. Okay, and it will go and bind to the terminator sequence, TER ETA or TER CB there, and it will block the replication by destabilizing polymerase 3. So polymerase 3 now further, it cannot move further, right? There is a destabilizing factor that is bound to that TUS sequence there, right? So terminator sequence, so TUS protein has bound there, so it cannot move further and it's blocked there, and that's how the termination in prokaryotes occur. Now this is not the end, okay, because we still have primers there. So primer has uh, the thing there, because these are the RNA primers, right? So it has to be removed, and it is done by either RNA-H or DNA Pol 1 in prokaryotes. Okay, now once you remove those primers, again you need to add the nucleotides, DNA nucleotides there, right? That is done by DNA Pol 1. So gap filling is done. Later, even after adding the gaps, the phosphodiester linkages are to be made after filling that gap. Okay, because that particular last bond cannot be done by Pol 1. So that is done by DNA ligase. So that ligates the fragment, and that's how you get the replicated DNA in prokaryote. So that's all about the DNA replication in prokaryotes like E. coli. Thank you. Hope that was helpful.